Good morning, everyone. I'm really pleased to be here at this ILSI symposium, where I'm going to talk about glycemic control and its impact and implications for cognitive function. My name is Louise. I'm the current president of ILSI Europe, and I'm also a psychologist from the University of Leeds. So this morning we're going to talk about glycemic control. It's useful to talk about different cognitive domains. I'd like to consider a kind of dose-response effect of glucose in healthy adults, and also then the impact of what happens to cognitive function in situations where one loses one's glucose control, particularly impaired glucose tolerance, so kind of pre-diabetes, and also then in type 2 diabetes. One of the predictive factors for the loss of glycemic control is obesity, and I want to focus a little bit on midlife. I'll also talk a little bit about how that's related to dementia and the role of stress in this. And I think ultimately what we're asking, the question we're asking is whether obesity or diabetes related cognitive impairments could be reversed by nutritional intervention. So I hopefully show you some positive evidence of this. I think it's really important, first of all, to think about different types of memory, because we talk about different cognitive domains, things like attention, reaction time. But one of the areas that's most easily influenced by glucose and glycemic control is memory. And memory changes as we age, but the decline in memory is greater in some memory tasks than in others. So we have a slight decline in what we call non-declarative memory. So that's doing things by rote or implicit memory. So if you learnt your timetables, you will always remember that 8 times 8 equals 64, for example. But problems with episodic memory are more common in mild cognitive impairment and in early stage Alzheimer's disease. So procedural memories then are knowing how to do something. Declarative memories are knowing that the President of the United States is now Joe Biden, for example. Semantic memories reflect our general knowledge, but episodic memories are our personal recollections. I remember getting married. I remember when I got my first dog or my first job, etc. So those kinds of memory problems, remembering what happened and when, are most common in mild cognitive impairment, and a decline in prospective memory, so remembering to do something, is quite common too with aging. So if we think about energy then, your brain sits in your cranium. It weighs about 3 pounds, so it's only about 2% of your body mass, but it uses 20% of the energy that you consume, and it uses it in the form of glucose and oxygen. So 2,000 to 3,000 pints or 1,500 litres of blood each day pass through the brain. So the brain is the most metabolically active organ, and that energy that we consume is used to drive neural activity. And as I said, we use 2 energy substrates, glucose and oxygen. We also can use other substrates such as lactate and ketone bodies, and other sugars such as fructose can lead to increasing circulating lactate, pyruvate and ketone bodies. So if we give people glucose, then there's a kind of an assumption that that might have a benefit to cognitive function. And there has been quite some evidence suggesting that there are benefits of giving small doses of glucose for things like information processing and attention, for working memory, executive function, and for long-term memory. And that there's a sort of a dose-response relationship, which is particularly evident for those declarative memory tasks. Glucose seems to facilitate memory if you consume around 25 grams of glucose, and that seems to be the optimal dose for most memory tasks for younger populations. However, we've got to bear in mind that the optimal dose may depend on age, individual differences in glucose metabolism, and the different cognitive domains that we're measuring. And what we've seen here from a study by Owens is that if we look at the area under the curve and immediate word recall, so this is remembering lists of words, what we see here is that in young healthy subjects with poor glucose regulation, they show an effect that's actually similar to that observed in the elderly. So if your glucose regulation is poorer, your performance is more akin to what we would see in older adults. Here's a nice study an old study now by David Benton, and he was able to give biscuits or cereals that were high GI in the green or low GI in the red. And what you see is a very clear separation of the glycemic response. So you would anticipate then that the performance would be better when the glycemic response was higher, if glucose was facilitating. What he actually showed was that the performance was best, at quite a long time after that glucose was ingested, and actually it was the low GI breakfast that resulted in the better performance. And the better performance occurred at the point where the glycemic response or the blood glucose levels had returned to normal. So it may well be that having a very high GI meal has a consequence for other metabolic processes. So you need insulin to remove that high glucose response from the bloodstream, and therefore a low GI breakfast is actually better for cognitive performance because it doesn't challenge the metabolism quite so much. We know too, when we do studies, that there is something called a second meal effect. So Tom Wolliver showed this quite some time ago, that what you eat in your first meal has an impact on your glycemic response to a second meal. And what we've been able to show in my lab is that the GI of the evening meal influences the glycemic response to your breakfast the next day, and it also therefore influences your memory performance the next day. And that's got lots of implications for how we do studies like this. Another thing that gets forgotten quite a lot is palatability. The same glucose given in 2 different meals that are different in palatability makes a difference to the glycemic response. So a more palatable meal will produce a higher glycemic response. If we think now about when glucose regulation goes wrong, before anyone becomes diabetic, they will go through a period of impaired glucose tolerance. And you can see here that that's actually quite prevalent and quite well spread across most of the world. We looked some time ago at the effects of glucose in relation to glucose regulation in a systematic review, because epidemiological studies had shown a clear association with cognitive function. So we wanted to look at studies where the participants were impaired glucose tolerant, they were in a pre-diabetic state, they were losing regulation, but they were not on treatment. They were predominantly middle-aged, and we actually found very few effects on cognition, but that was probably because most of the studies at the time had used very insensitive tests. So they'd used dementia-type tests, which wouldn't be suitable for normal cognitive function measures in middle-aged participants. So we then went on to do a study where we recruited women, ostensibly for a breakfast study, so we didn't ask for people who were impaired in their glucose tolerance, and they were aged 30 to 50 years from the general population. So we screened them and we gave them an oral glucose tolerance test. What we found was that around 1/3 of the sample showed impaired glucose tolerance. So here you can see the blood glucose profiles, quite different. In the red, the impaired glucose tolerant, and compared with the normal glucose tolerance. Normal glucose tolerance means that you return to baseline within 2 hours of your glucose load. When we looked at cognitive function in those participants, we saw in terms of the number of words that they forgot during a memory test, which we call retroactive interference, that those with impaired glucose tolerance in the dark bars showed greater cognitive fatigue. They also showed poorer delayed recognition memory and poorer recall of verbal material. So that suggests a deficit in hippocampal function. Across all the studies, all the measures that we used then, we actually found that the impaired glucose tolerant group were impaired on 12 out of 27 cognitive test outcomes. So that's way more than you would expect by chance. And they were impaired on verbal learning, on word recognition, on visuospatial learning, and on psychomotor and spatial memory tasks. So these were subtle impairments in prolonged concentration in women who were ostensibly healthy and middle-aged. Now, what's important is that a healthy diet and exercise can actually halve the risk of developing diabetes in people with impaired glucose tolerance. So let's think about when glucose tolerance gets even worse and we move on to diabetes. We know that the prevalence of diabetes is high. This is data from the UK, and it's rising. So we had around 3.8 million people diagnosed, and we estimate around 1 million people wandering around with undiagnosed diabetes in the UK. We estimate that by 2025 that will have risen to 5 million people, and 90% of these have type 2 diabetes. What we're also seeing is an earlier onset. So while there's a genetic predisposition, and that accounts for some of the high rates in places like Bradford, Birmingham and Leicester, where Afro-Caribbean and South Asian populations have a higher rate of diabetes and a higher risk of diabetes, Bradford has the highest prevalence in the UK. Around 1 in 10 of the population has type 2 diabetes. So it's a significant problem. We know from the literature that type 2 diabetes is associated with cognitive impairments and an accelerated rate of cognitive aging. And fMRI studies have actually shown that the hippocampus, that area of the brain where memory takes place, is reduced in volume, with implications for that memory performance. But obviously, mostly people get diabetes as they get older, so some of these effects are confounded by the cognitive effects of aging. What we see in middle-aged adults then with diabetes are modest decrements in specific cognitive functions, whereas when they get older, you see older adults with diabetes have widespread decrements that impact more cognitive functions and affect things like processing speed, attention and executive function, as well as memory. This is an example from a study that we did in my lab, where we looked at type 2 diabetics and we compared them to normal glucose tolerance adults who were matched on age, education, depression and IQ. And what you can see here is that the type 2 diabetics made fewer correct responses than the NGT group at all levels of this task, except the very easy levels where they only had to make 2 or 3 decisions. Once it got into more complex, you see that drop in performance with both age groups, but it's so much worse in the type 2 diabetics. They also showed deficits in verbal memory and psychomotor skill and executive functions. So obesity then is one of the precursors of type 2 diabetes, and we know from studies in the US that obese adults tend to perform worse than normal weight adults on tests of learning and memory, and also frontal lobe tasks that engage the frontal part of the brain. And that potentially that impaired decision making that the frontal lobe does might also contribute to some poorer dietary choices. So it could be a circular behavioural reinforcing activity. What's important then is not just how fat you are, but where you carry that obesity. And so waist-hip ratio is quite important. So having a high waist-hip ratio and therefore being more of an apple shape than a pear shape is more negative in terms of risk of metabolic syndrome, and we think also cognitive deficits. So when we look at waist-hip ratio and hippocampal volume, you see that as it increases, hippocampal volume decreases. And there's been some implications that increased visceral fat might have something to do with cognitive decline. What we've shown in our lab is that increased waist-hip ratio was associated with poorer spatial recognition memory in middle-aged men when we exposed them to stress, and they also had greater cortisol response to that stressor. Now, if we reverse obesity, the most drastic way of doing so is bariatric surgery. And there's been a recent systematic review that looked at what happens to cognitive function in studies of people that underwent bariatric surgery. And what we can see here is that those individuals on the whole had a very rapid improvement in memory, in executive function, and in cognitive control. But you don't see that pattern in individuals who have a family history of Alzheimer's disease, which may be more related to genetic predictors of AD. What we see in patients with type 2 diabetes is that the relative risk of vascular dementia is 2.5 times greater, and the relative risk of Alzheimer's disease is 1.5 times greater. So having type 2 diabetes increases risk of dementia. So can we improve cognitive function? Can we reverse some of these deficits through weight loss? A number of studies have used the DASH diet and shown that hypertensive on the DASH diet, where it's coupled with aerobic exercise and reduced calories, actually improve on multiple measures of cognitive functions. And losing 14 kilos over 1 year, which is around 1 kilo a month, which is kind of a little bit a nice steady weight loss, is also associated with improvements in working memory. So I think what that tells us is that obesity-related cognitive function is partly reversible, but we don't really understand the mechanisms and the physiological processes for that. This is a really nice Japanese study in octogenarians. So this is Japanese adults aged over 80 who underwent a 2-year intervention. So they did exercise 2 to 4 times a week, exercises like Tai Chi, and they increased their dietary fibre to over 30 grams a day. Amazingly, the majority of the group had normal glucose tolerance. Some were in that impaired range, and a smaller group were type 2 diabetic already. What we can see here is that a 0 means there was no change, a minus means it went down, a plus sign means it got better. What we see here is that fasting plasma glucose went down in the diabetics and those with impaired glucose tolerance. It didn't change in the others. Same happened for the oral glucose tolerance test, and same happened for HOMA, which is a measure of insulin resistance. The mini mental state didn't change in the first 2 groups, but improved in the type 2 diabetes. And as I said before, that's quite an insensitive task. So you have to have quite strong impairment to show a deficit on that task in the first place. The same pattern was observed for the dementia scale. And then the tests that I showed you earlier, things like delayed recall and block design, improved in those groups. So diet and exercise, then dietary fibre, which ameliorates big glucose excursions, improved cognitive function, probably because it improved glucose regulation. So it seems then that we don't want to wait until we're 80 to try and improve our glucose regulation. I would argue that the midlife period is a really critical period to prevent type 2 diabetes, to make sure people continue to have good glucose regulation and therefore to prevent cognitive decline. So in conclusion then, I think the best we could sum up by saying that it's very clear that type 2 diabetes is associated with poorer cognitive function, but it's domain specific. It needs to be detected by sensitive cognitive tasks, not global function, not IQ, not things like the mini mental state examination. The cognitive effects that I've shown you are evident, but they're less severe in people with impaired glucose tolerance, and obesity is associated with that poorer cognitive function. Obesity is also, or cognitive function impairments are also exacerbated by stress. And we know that people under stress expand their waist-hip ratio, and stress can lead to the laying down of more abdominal obesity. So the causal factors here are glucose regulation, insulin resistance, and some other factors. Reducing obesity at midlife would be beneficial, and it's very important, I think, that we do those things in midlife in order to preserve cognitive capacity in aging, because that's the thing that enables us to live independently for longer. So that's my group who've been involved in this research. There's a nice study, a nice review published in the Proceedings of the Nutrition Society, in case that's interesting for people. And there's also a book on brain health and cognitive performance, which covers some of these topics. Thank you very much.